Cure, Connect, Transform. There is no greater embodiment of Mayo Clinic's plans to revolutionize healthcare than the Center for Individualized Medicine. We discover, translate, and apply new individualized medicine advances in patient care. Using genomics and multiomics could lead to vast advancements in knowledge of the causes and risk factors for many diseases. Our researchers and clinicians work together to accelerate discoveries in 4 strategic areas. Simplify and scale. In Genomics in Action, we work to transform the practice by providing the knowledge, tests, and tools needed to make it easier for clinicians to integrate genomics into routine patient care. Individualizing cures with genes, bugs, and drugs. Cutting-edge therapies being developed with high-definition therapeutics are driving precision medicine science beyond diagnostics to provide personalized cures. Connecting data to practice. Our exciting work with Omics Data Platform is centralizing genomic and multiomic data into a single repository, which will allow for the most individualized medical approaches possible. A multi-omics future. In Beyond DNA, we are advancing patient care by using molecular information from the microbiome, epigenome, exposome, and more.
The Center for Individualized Medicine and Mayo Clinic are in a unique position to fuel new discoveries to improve care for patients powered by sophisticated research and artificial intelligence. From providing the best individualized care to addressing the world's most challenging healthcare problems, Mayo researchers here are relentlessly pursuing discoveries that deliver hope and better health to people today and for generations to come. Thank you.
Good morning, everyone. I guess good afternoon now. My name is William Palmer. I'm the Associate Director for Education for the Center for Individualized Medicine in Florida. I'd like to welcome you to the Florida Center for Individualized Medicine Grand Rounds. The CIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation, and to demonstrate how individualized medicine is being translated into practice to meet the current and future needs of our patients. It's my pleasure today to introduce our speaker, Dr. Dean Jones. Dr. Jones is Professor of Medicine in Pulmonary Allergy and Critical Care, as well as Biochemistry at Emory University, and he's the Director of the Clinical Biomarkers Laboratory. His studies include redox biology and medicine, and he has research programs in areas of redox systems biology, clinical metabolomics, and environmental health. In recent years, Dr. Jones has developed an Exposome research program, building upon the metabolomics and mass spectrometry programs for affordable, high-throughput environmental chemical biomonitoring. Dr. Jones is recognized for his research in oxidative stress, environmental health and toxicology, mitochondrial mechanisms of cell death, and antioxidant disease. He's received the Albert E. Levy Research Award, the Science and Humanitarian Award of the Oxygen Club of California, and is a member of the Emory Millipub Club. He's received the R. Wayne Alexander Research Career Achievement Award within the Society of Toxicology and Mechanism Lifetime Achievement Award. He's also received the JARO Science Advancement Award. Today's presentation is titled The Development of Exposome Research Methods to Support Inclusion of Lifelong Environmental Exposures into Precision Medicine. He will provide a background into exposomes as a complement to the genome, as major factors impacting human health and disease, as well as describe the progress with powerful new omic scale clinical chemistry analysis, which are poised to revolutionize health predictions and precision therapeutics. Realistic considerations of obstacles to implementation and time frame cannot be predicted. If you want to learn more about the exposomes and hear Dr. Jones' deeper dive into this topic, make sure you register for the Center for Individualized Medicine Conference that's happening next week in Rochester from November 2nd to 3rd. And this will also be available virtually. The link that includes in the chat will include a discount code for the course. Throughout this talk, please use the Q&A function in Zoom to submit questions. I will interject those questions into his talk as appropriate and will answer the remaining questions at the end. For those of you who have not claimed continuing education credit, a message will be sent out shortly in the chat. So please join me in welcoming Dr. Jones.
Okay, thank you. Now I have to try to remember what I'm doing, sharing the screen. It's a pleasure to be here, and try not to be overwhelmed by what I'm going to talk about, because it's overwhelming no matter how you look at it. First off, I have no disclosures or financial conflicts of interest, and I have many funding acknowledgements, and I'm not going to go through those, but I've had the good fortune to have support by many federal agencies to conduct this research. You should also feel free to ask questions throughout, just through the chat, and those will be relayed to me. I can, the purpose really is to exchange information, and so I don't need to cover every single slide. What's most important is that I'm clear and convey the central aspects of the exposome. I'm going to have this organized into three components. I'm going to talk first on the exposome research paradigm. Recognize that we talk about the exposome research because that's where the main focus is today. But the exposome is very broad, and it's not that it hasn't been included in medical practice. It has, very simply, things such as alcohol abuse, such as cigarette smoking, and so forth. These are parts of the exposome, and they are well covered in terms of our usual clinical practice. But what we want to do is we want to push it beyond that. And so I will go through what we're doing and the methods that we've developed. I will then move on to talk about how we're really pushing the limit on ability to measure especially the chemical exposures that we have. And then finally, I will talk a bit about the introduction of these concepts into complex disease etiology and how that's being approached. So if we go back 40 years or so, we recognize that we already knew about environmental contributions and what is the relative contribution of different factors to cancer development. And this environmental epidemiology really is the background for the whole concept of the exposome, because it was the former director of IARC, Christopher Wild, who really took the next step. He said that with the completion of the human genome, that it was really necessary to look forward to really understanding the exposome, that complement to the genome in terms of contributing factors for human disease. And so what we now recognize in terms of the contributions is that, in fact, the environmental contributions are probably greater than the heritable contribution, or at least the environmental exposures in combination with the host genome. And so the more recent depiction of the relative contributions to disease is shown here, where the genetic contributions alone are really only a small fraction, or not a small fraction, but only a fraction of the total risks. And of course, this doesn't have infectious disease in it. That this is really the non-infectious disease component of disease. But what you'll see is that there's a vast portion here that is unknown. And of course, when it comes to things like the diet is so variable that is difficult to define in a precise way. So we outlined sort of a primer for the exposome for high school educators several years ago. This was done by Christine Dennis, who was a Ph.D. student with me at the time. And this visualization really captures the crux of the matter, and that is throughout lifespan, we have exposures. And those exposures can have long-lasting impact on our health and disease outcome. And so that's really what the exposome research is all about, is really mapping out those environmental exposures contributing to disease. Now, of course, there's major challenges. One is that the humans live a very long time. So if we want to be able to predict something over a life course, well, we really have to be very creative in how to do that, because we're not going to have answers for decades for some of the important questions. There are, of course, many types of exposures, and these are highly variable in terms of exposure histories. For instance, even things such as cigarette smoking can be very intermittent. It can be chronic. It can be short duration. It can be almost lifelong duration. And so there's tremendous variations in specific exposures. And the real complicating factor is that there are so many exposures that one has to consider co-exposures, the sequence of exposures, and also the interactions of the exposure with the host genome. So it's not a trivial matter what we're trying to address here, but the concept is that we will be able to do it. We just have to be clever enough to figure out how to do it. And so this was from one of my presentations already about a decade ago. I was giving a lecture in New York City just after Sandy came through. And what the point I wanted to make was that if we look at what the Hurricane Forecast Center did in order to be able to predict hurricanes, they started off really very poor. And the way they got better was that they put in the necessary resources to capture the information that they needed to predict. And then they developed lots and lots and lots of different predictive models. And they really learned from the failure of those models. And unfortunately, we don't do that much in medicine. We focus on the victories and not the failures, but it's really the focus on the failures that we need to make in order to be able to advance things, to be able to correct the problems. But anyway, the concept is that what we want to do is we want to be able to better predict risk of disease, timing of disease onset, and the intensity of disease impact. And if we could do this, we could obviously make the healthcare system much more efficient, and we could also better empower individuals with their own health management. So the needs are considerable, because we start with, if we have high dose chronic exposures, such as long-term cigarette smoking, that's relatively easy to evaluate the risks, but it's still not completely simple, obviously. But if we think about low dose chronic exposures, then we have to be, we are limited basically by how sensitive our assays are for being able to detect and quantify those chronic exposures. But it becomes far more difficult when we have intermittent exposures. And this is now limited by the frequency of testing. So if someone has an exposure for 10 months out of the year, but that has nothing, and that's not occurring when they're coming in for their annual physical exam, if you do tests during the physical exam, you may not pick up that exposure at all. So so this is one of the difficult problems that we have to face, is how do we get better coverage for intermittent exposures. And then we have some that are even more difficult, and that is when we only have transient exposures. So we've done a lot of work with the Department of Defense on trying to evaluate health risks from military exposures during deployment. And what we conclude is that we really have to have detection systems in place to be able to measure these exposures, or we must have means to measure the biological response to exposures. And so in terms of the big picture on the exposome, this last concept here, means to measure biologic response to exposure, led Gary Miller and me to refine Christopher Wilde's definition of the exposome. So in the original definition, it was really just the lifelong exposures. But from this example of biologic responses to exposure, it means that we really have to include within the exposome not only the cumulative lifelong exposures, but also the cumulative biological responses to those exposures. And so that's really the bigger picture as far as the exposome contribution to health outcomes. So in terms of simple solution, okay, this was my naive concept a decade ago. I was the director of our general clinical research laboratory, and I had a very simple solution for this universal exposure surveillance. And that was, we just simply replace standard chemical profiling, which we use routinely, like for instance, in annual physical exams. If we just simply replaced that with a high resolution mass spectrometry analysis, we would get most of that information that's already available from those current clinical tests, and at least the chemical component of it, not the immunological components, obviously. But it would also then give us that information and also give us the information on exposures. Any chemicals that were within the sample that could be measured would then give some background measure of what that individual was exposed to. So the concept was that we can't do it today, but if we started this today, we could collect the data so that future scientists could go ahead and start developing models and developing these predictions. And then our children and our grandchildren and great-grandchildren would then have the advantage of having lifelong measures of exposure, and therefore we would learn which of those exposures are really most important in adverse outcomes. So that was a concept. That's in this article, Sequencing the Exposome, a call to action that I wrote several years ago. But anyway, this has not made, we haven't made much progress on this. Nobody has really made much progress on this. However, there's now hundreds of publications showing feasibility of this. It hasn't been brought together, and hopefully that's one of the things that the Center for Individualized Medicine will be able to accomplish, because this is really, really needed. So that was the first part, just the exposome research paradigm, the concept that we can measure exposures, and we want to incorporate those into our health management. Health prediction, health management. So I'm going to now move on to the analysis of this. I have a few slides on mass spec, not much, but it's just really to emphasize that there are lots of experts out there in analytical chemistry, in clinical chemistry, who are really leading the way in terms of development of new analytics for the exposome. So where I started on this was now back in 2003 with the concept. At that time, we were calling it personalized medicine or individualized medicine, and now it's usually termed precision medicine. But in any case, the concept was really that the age-old concept of disease, that there is a specific cause. And this goes back to the central dogma that DNA is how our information is stored, and it's converted to RNA and to protein. And then with only about 40 essential nutrients in our diet, we're able to produce all the metabolites that we need and produce energy and do everything that we need to do for complex living systems. And so the medical aspect then is that the disease disrupts at a specific site, and therefore our value of metabolomics is really for biomarker discovery. In fact, I'm director of the clinical biomarkers laboratory, and that's the concept, is that we want to have specific biomarkers to be able to detect the site of disruption in disease etiology, to guide the therapies and to monitor the efficacy of treatment or management. And then ultimately, we want to be able to develop biomarkers to predict health risks and prevent disease. And so from this perspective, metabolomics only needs to measure some key targets. And so our estimates were at that time only perhaps 200 to 300 key metabolites would give you all the biomarkers you would need for the major diseases and their risks and so forth. So what we found already in 2010 was that with a single mass spectrometry analysis, we could measure metabolites in virtually every metabolic pathway, human metabolic pathway. It was really quite remarkable. And so the concept was, this was really behind that concept, that we should just replace the usual clinical chemistries with a high resolution mass spectrometry analysis, which would give us all that information, plus a lot more. And what we also realized at that time was that that analysis gave us far more than just those core metabolites. It gave us measures of what was in the diet that the individuals were eating. It gave us information on the microbiome products. Of course, when we eat, the food first goes into our gut, and our intestinal microbiome converts those nutrients to many different products. But also we get measures of dietary supplements and pharmaceuticals that individuals are consuming. We get measures of commercial products. These are personal use products, for instance. And we also get measures of environmental chemicals. So that single analysis gives a very broad spectrum of the human chemistry, the human biological chemistry, what is in whatever sample that is measured, whether it be plasma or serum or whole blood or urine, saliva. Virtually any biological sample can be analyzed, and even biopsy samples can be extremely valuable. So what we found from this work when we started doing bioinformatics was that that buried within that chemical, all the chemicals that were being measured, there was information on the association of chemical exposures and the metabolic response. And so this was really eye-opening, because it gave really the foundation for exposome research model. And because with a single sample, you not only got a measure of the exposure, but you got a measure of the biologic response. And so that has been really developed, and the first complete study establishing this exposome research paradigm was done by Doug Walker when Doug was a graduate student working. He was actually got his Ph.D. at Tufts in environmental engineering, but he did much of his Ph.D. research here at Emory, working with myself and with Gary Miller. And he's the one, as a graduate student, who did the foundational work establishing this research paradigm. First publication showing the entire linkage of external exposures to internal dose, to biologic response or metabolic response, and to health outcome was done in an occupational research study with Nat Rothman at the National Cancer Institute. Samples that they had collected and already documented the exposures. And so this research paradigm has been fully validated in that sense and established, and it's been very useful. There's now hundreds of papers based upon this paradigm that we can link external exposures with internal doses, and not necessarily of the precursor, because it can be bioconverted within the body, but with biologic metabolites in the body, and that those can be linked to metabolic responses to the exposure, and then those can be linked to biomarkers of risk of outcome, as well as adverse health, beneficial as well as adverse health outcomes. Okay, so covered a lot of territory at this point. I have to jump back to history. So this, the reason I like this copy of paper by Ron Estabrook in a National Research Council publication, 1981-82, is he starts off with the first sentence referring to my Ph.D. mentor, Howard S. Mason, introduction of the term xenobiotic to identify substances foreign to the metabolic networks of higher organisms. And so the concept here is, this is really broadly what we're talking about with exposomics, is we're talking about exposures to chemicals that we don't normally see in our body. And so these are termed xenobiotics. And already at this time, Ron had gone through the literature and gone through all the production records and so forth, and had estimated that there were 60,000 chemicals of important commercial use. And he defined that as use of, by greater than 1 ton of the chemical was being used per year. And if we go into the literature today, what we'd find is that there are about 100,000 agents that are registered for commercial use with the US EPA. And if we want to think about this critically, of course, we've mostly phased out, extensively phased out the BPA use in this country. But at its peak, for many years, there was approximately 400 metric tons of BPA produced per year. And of course, what one must think about in terms of commercial chemicals is that they are not necessarily pure. Sometimes they're 99% pure, but sometimes they're only 90% pure. And so what that means is that for every high production volume chemical, there are going to be metric tons of uncharacterized impurities. And so the concept is that there's vast amounts and vast number of these chemicals. Now, to make it worse, we have between 75 and 100 different enzyme systems that modify foreign chemicals. These are xenobiotic metabolic enzymes, and their purpose is to make the chemicals more oxidized and therefore eliminated in the urine. But what this does is it creates a very large number of other chemicals, because we can have sometimes 5 or more of these modifications on the same xenobiotic. And so if we just do a calculation, a simple calculation, if there's 60,000 chemical exposures and an average of 5 metabolic products, then we're talking about 300,000 chemicals. And if we look at the literature, we find that humans are not alone in being able to catalyze these xenobiotic biotransformations, but in fact, there's over 1,200 bacterial enzyme reactions that are known. And of course, our intestinal microbiome contains large numbers of chemicals. So what we're dealing with is a major problem, and we call it a mega scale problem, because it's not a small number of transformations, it's a very large number of transformations. We actually refer to this as the dark matter of the exposome, because we don't even know how many of these modified chemicals there are out there. And so from my standpoint, this is the most traumatizing aspect of the whole subject, is this dark matter of the exposome. Because if we go to the literature, probably everybody who's listening is aware of the persistent organic pollutants, which bioaccumulate and therefore have been basically phased out and are no longer used commercially, but they also biomagnify. And what this means is that these persistent organic pollutants, the POPs, oftentimes were unintended byproducts, like the hexachlorobenzene, and many of these were really unintentional byproducts of manufacturing. And what we now know is many of these are endocrine disruptors. That is, they disrupt our endocrine systems in terms of reproduction and development, as well as interrupting cognitive functions and causing cancer and so forth. And to me, the traumatizing aspect is that, well, what happens if the contaminants in the environmental chemicals or commercial products, or their biotransformation products or breakdown products, such as what we use every day in our shampoos, skin products, water purification systems, even things like our fluoridated toothpaste. What happens if those are converted to stable products, which bioaccumulate and are endocrine disruptors? We basically have no way to predict it. We have no way to measure it. And yet it's a legacy of our current society. And so this is something that from my standpoint, we must focus on the exposome, and we must learn what is important. Obviously, most is probably not going to be important, but some of these are going to be important, and we need to identify those.
Dr. Jones,
yes,
there's a question that came in regarding, as you're commenting on those exposures, mental stress as a risk factor for disease, and how could that play a role? Are there any proposed ways to measure or reference that?
Yes, actually, we have a couple publications on chronic stress, and it's absolutely true. Some of the biochemistry has been worked out for some of those. For instance, some of those breakdown products, oxidation products of tyrosine and tryptophan that have been recognized to contribute. So I think even though it's not a chemical exposure, stress is not a chemical exposure, the biologic response is producing chemicals in an adverse way, and those are contributing. I won't pretend that we know the answer to all these things. I think it's certainly something that we and others have worked on, and it's something that is very important. And if we can get good assays for how to control it and interventions, I think that's going to be very critical. But yes, that's a very important point. Psychosocial stress is a big deal, and I think a major contributor to adverse health. I mean, the data show it. It's a major contributor. So hopefully that answers the question. Can come back and discuss this more. So jumping back to the environmental chemicals, one of the challenges is that environmental chemicals are present at relatively low abundances. And so if we look at this plot from one of Steve Rappaport's papers now many years ago, where he quantified blood levels of drugs, dietary components, endogenous metabolites, and environmental chemicals, he concluded that most of the time the environmental chemicals are 3 to 5 orders of magnitude, meaning 1,000 to 100,000 lower concentrations. So they're just a lot more difficult to measure than the higher abundance exposures. So what we did in terms of working on this back initially started on, we got our initial funding back in 2007 on this, and our progress was really made early on in terms of boosting the number of chemicals that we could measure. And this in 2012, we had achieved about 20,000 chemical signals. And I called that the threshold for precision medicine, because there are approximately 20,000 genes. And so when we can get to the point where we're getting 20,000 chemical signals, then we're on par in that omics level to the genome. Of course, the genome has so many different variations in terms of the structure, it's a lot more than 20,000 variations. And obviously there's going to be a lot more than 20,000. But just to think about things, this 20,000 number is pretty much where we are. We can get a bit more than that. We can maybe get 100,000 signals on a routine basis. But 20,000 takes us a long way towards dealing with the, with the major factors in the exposome. And just a quick overview of some of these from our, we've just completed a compound identification discovery core with the Metabolomics, NIH-supported Metabolomics Consortium. And in this, we mapped out the metabolites from about 2,500 chemicals. So we generated about 10,000 xenobiotic metabolites, and you can see the spectrum of chemicals that this covers. And so we are now getting the ability to measure a very broad spectrum of environment, exposures of different types: agricultural chemicals, natural products, household chemicals, pesticides, personal use products, and so forth. So there's a lot of those that are out there, and we're now nominally, I'm going to say that nominally because the methods aren't really fully perfected at this point, but we're able to measure a very large number of these in human samples, human and other biologic samples. This is, I think, my only mass spectrometry slide, and so it's mostly intended to let you know that unless you are specifically interested in as an analytical chemist, you probably want to leave it to the clinical chemists or analytical chemists and trust them to do a great job, that's what I do. But there are three main types of mass spectrometry that are used for exposome research. The most common is liquid chromatography coupled to mass spectrometry. They're on the top, most common for metabolomics here. The second for environmental chemicals is this gas chromatography coupled to mass spectrometry. And then for metal analysis, the ICP-MS is most commonly used. I'm not going to go through that. There's lots of detail with regard to how things are done. And, just you have to trust that the chemists know what they're doing, and we have to push them for doing it better. And so that's what we're doing. And this is one of the papers we recently had that Shen Hu did on using the gas chromatography, showing that they can quantify over 1,000 different environmental chemicals, and this sort of gives a spectrum of some of those that can be measured by this method. So coming back to the central concept, heritability accounts for only a small fraction of human disease. Key need is for routine omics scale measures of environmental chemicals. We need to have them in comparable sensitivity and coverage to genomics. And from my perspective, we need to get these into routine healthcare, because if we could, then we could begin to address the cumulative effects of the different exposures. So in this inverted pyramid here that we had in a paper now several years ago on the million metabolome, main point on the main limitations that cost limits the analyses to most top priority chemicals. And so if you go into the NHANES and to EPA, for their routine surveillance, they aren't even getting up to 2,000 with a broad coverage, simply because it's too expensive in a targeted way. The capabilities now, as indicated with the omics research, is that we are at this level of being able to measure tens of thousands of signals. Now, keep in mind that we haven't identified tens of thousands. We can only measure tens of thousands. So there's a lot of chemical structural elucidation that's needed there. And it's only an interim solution at the omics scale, because ultimately we can predict that we're exposed to over 1 million different chemicals and metabolites. And so we have a long ways to go here. And I think the point is that, being realistic, we can go to the omics scale. And a lot of these issues were discussed in this paper by the group several years ago, where we framed out the concept of how would we go about the million metabolome, actually doing this at a mega scale. Okay, the last few minutes, I want to talk very briefly about trying to put this exposome, already an overwhelming concept, trying to put this into the context of complex disease etiology. And what I must point out, although I don't like to, is that multimorbidity is the most common disease condition. And when you get to my age, an average person has 3 chronic health conditions. So we're not dealing with one disease. We're dealing with most people having multiple disease. And if you're a youngster over there and you think, well, I'm young and don't have any of those, well, just think about it this way. You look at this plot, all it means is that you are pre-disease. You just don't necessarily know what you're pre-disease for. And thus, the information is still relevant to you, even though you're young and healthy. So the way this is being handled, has been handled now for several years, is this aspect of network medicine. The concept is that we need to address those interactions of disease and causal mechanisms. And in so doing, we will get a better understanding of who's at risk of what, and also then what comorbidities are likely to be seen. Of course, this is, in a major way, this is already in practice in individualized healthcare. But the point is, if we can do it better, we want to do it better. And what we need to do is bring the exposome into this, is what I think. So just for a quick refresher on network theory, what we're really doing is we're visualizing relationships. And so these are typically discussed in terms of nodes. Those are, for instance, measured entities, and edges, which is the relationship of different nodes to each other. And hubs are nodes that have a lot of connections, or really important in the overall structure. And there's lots of different types of networks. What we're discussing, these are usually in terms of relevance networks, where we're trying to connect biomedical and clinical entities. What we do in our research group is we do a lot of proteomics by metabolomics, or metabolomics by transcriptomics, or metabolomics by clinical outcome. So we can do all sorts of different types of associations and map them out using these network visualization tools. So in these visualizations, typically the larger the individual component, that means the more connections or the more influence that component is in the overall network structure. Now, if you look at the exposome paradigm, you'll see right here in the middle, that's what we're looking at, is we're looking at internal environmental chemical exposures, or internal dose, and their associations with metabolic responses. And again, I recognize the time, and so I didn't put in a lot of examples here, but I did want to just show that we can now do these kind of links throughout all of the omics space, because we can connect the genome with the epigenome, the transcriptome, the proteome, the metabolome, and then pull all of those in with immunologic measures, with the microbiome, and with the exposome. And this is just quickly what you see. And although this looks overwhelming, it's actually a great simplification, because, for instance, on the right here, this complex network at a genome scale, what that tells you is that within these different communities or these clusters, these components are strongly interacting, they're highly interacting. And therefore, we don't have to measure all of those. If we can get the right biomarker for that entity, then we actually can greatly simplify that overall complex network. This is an example. This is a microbiome metabolome interaction study of human bronchoalveolar lavage samples from healthy individuals who have chronic HIV-1 infection. And so what this showed by doing the microbiome analysis and doing the interaction of that microbiome analysis with the network analysis with the metabolome, what it showed was that the 4 species or 4 groups of organisms that cause opportunistic infection actually form the hub of the metabolic network structures, even in the healthy individuals. And so it really, even without knowing any of the metabolite identifications, just seeing the structure, it reinforces what we know about the opportunistic infections. And there are many examples of this. We've done several studies on air pollution, looking at this is one that we did with Beatrice's group. And what it showed was that we could take those very complex networks and focus them down to individual, well understood biochemical processes, such in this case with the fatty acid oxidations, the linoleate and arachidonate oxidation products, as well as antioxidant systems, which were also linked in these complex networks. This is another air pollution study, and it gets more detail, but what you can see is you can zoom in and zoom out on these network structures to get a lot of biochemical understanding. And also, ultimately, we're going to be able to use this to be able, I believe, to intervene to reduce the adverse impacts of air pollution and other exposures. This is one on mitochondrial function. We collaborated with Victor Darley-Usmar on use of platelets to measure mitochondrial function in humans. And so we take human samples, isolate the platelets, and can perform the seahorse assay on mitochondrial function, and then do a metabolome-wide association study of those respiratory characteristics. And what we find here is in this center of the reserve capacity, very interesting, highly dependent upon the xenobiotic content and the phytochemical content of those platelets. So clearly there's a biologic response. We can measure it at an individual level on humans. And in this, it's not available clinically at this point, but this is an assay that definitely could be developed. And we've also done work, this is by Shin Hu, where we've done just survey of literature linking exposures to metabolic pathways and then disease outcomes to metabolic pathways. And what this has shown us is that we now have a way to characterize the functional reprogramming from long-term environmental exposures. And I think this is my next to last slide. Hope so, because I'm out of time. The other aspect of this is a study that we did in a non-human primate where we were comparing the metabolic structures of young individuals to old individuals. And what this shows is that from this metabolic network structure, again, just done on individuals, we can see that on the left, there is a very, very different metabolic network structure for the young female compared to the old female. And this contraction of these network structures is a sign of rigidity or flexibility in the metabolic responses. And basically, it's showing that that even in an individual, we can evaluate health in a very, just in a network sense without actually knowing any metabolites at all. Of course, some of these are labeled in here just for because we did that, but it isn't necessary in terms of the visualization. So that's where we're heading, is exposome medicine, and that is the use of exposome measures to aid diagnosis, management, prevention, and cure of disease. Still optimistic at this point, but I believe we're headed in the right direction. Summarizing, I talked about the exposome research paradigm, where we can link external exposures to internal body burden, biologic response, and phenotypes of disease. I talked about mega scale analysis, where we are. It's not easy, but we're at least at an omics scale now, and we're moving forward in development of new methods, and also what we'll have to do is use those in development of disease prediction models. And finally, we just collect the data. I believe we can trust that the future scientists will be able to deconvolute these exposures to be able to understand and better predict disease etiology. So I think I ended up pretty much on time. Lots of people in my group, and we've collaborated with so many groups across the country that I couldn't really legitimately list all of them and be fair. But obviously very active group, and lots of fun, and I appreciate having the opportunity to talk here, and happy to now answer questions.
Fantastic. Thank you, Dr. Jones. We've got a number of questions coming in. Just to get started, can you comment on the relationship between epigenetics and metabolomics?
So not a whole lot of literature on that. There are a few papers. What these are showing is it's actually very similar to the genome by metabolome interaction studies. You have some hot spots. You have some things that are identifiable in terms of specific changes in the epigenome that are linked to specific metabolic changes. When you have a complex system, it's hard, and you're only looking at 2 of the omics spaces. You don't know exactly what is intervening. So in the epigenome, you have the transcriptome, but then the transcriptome is impacting the proteome, and the proteome is interacting with the metabolome, and the metabolome is feeding back on the transcriptome and the proteome. So in the network world, it's always more complicated, but absolutely, if you go into literature, we've done a study on the epigenome in cigarette smokers and in the basically placental epigenome, and then the associations with the metabolome. And very clearly, those associations are there.
Can you comment briefly? You had made a mention about heritable factors and their risk to disease as being a fraction of that. Something like late onset Alzheimer's has a heritability risk between 58 to 79%, the number that's thrown out. So can you just comment on that balance?
Well, yes, that's going to be a complete spectrum. And the challenge is going to be taking it to the individualized level, because you don't want to be introducing unreasonable fear in individuals, as well as you don't want to make people be cavalier about their risks based upon their genome. So we just have to be careful there. We don't, it's clearly a balance that has to be, that we have to pay attention to. And this, at this point, we have to tread very carefully, just simply because of the bioethical issues. It's unreasonable either way to be wrong. And so we know there's a relationship. We know that both can be important, and we just have to be cautious in terms of where we are.
Fair enough.
And if you're a youngster, absolutely put the effort in there to do the science and get it so that the medicine is better.
Can you comment, there's a mention of 100,000 of metabolites needed for one study. So assuming that's needing hundreds of thousands of samples. Are there any rule of thumb guidelines for these studies that integrate the epigenome, microbiome, metabolome regarding the numbers of samples?
So the answer is there's not really a rule of thumb. What I suggest is that ultimately we want to go to N of 1. Okay, so individualized medicine means N of 1. So one can't really just say that you have to have a certain number of people, because, you know, Mike Snyder had a whole paper on himself in Cell. So, you know, it's completely okay to study one person if you're doing it in enough depth. But for rule of thumb, if you want any sort of generalizability, I think that you just don't want to be less than about 20 individuals. And I usually say 40 individuals is safer. But then you see you're going to have all sorts of demographic factors, age, sex, race. You know, you're going to have so many other factors that to be truly generalizable, you have to go to a population-based study. So that's the exposome epidemiology.
We'll do one final question. Are these all measures of blood? Are we doing any accumulation or exposure to specific organs or cell types, et cetera?
So most of the data is from readily accessible tissues like blood measures, plasma, serum. There are a lot of studies on urine, some on saliva, but all biologic fluids have been, as far as I know, have been measured at some point. We measured the fluid in, I can't remember, in the forelimb joints of mice at one point. Someone collected them. So it's possible to do very small samples, but the concept of organ measurements is something that's really just now developing. And we have a couple of papers on that recently. But yes, in terms of liver measurements, lung measurements, adrenal measurements, the different organ systems do collect environmental chemicals differently. They do accumulate. And at this point, and we've linked them to metabolic variations, but at this point, that's a very understudied aspect. Needs a lot more work.
And one rapid fire question. Do you have a standard panel that's published recently, either epigenome or metabolome?
Well, for a standard panel, our most standard that has been published was in Nature Communications in 2000. No, I guess it was that. It was in an analytical chemistry article in 2020. We had about 500 chemicals, metabolites that we measured. The environmental chemicals, we have that most complete would be in Shen Hu's paper that I mentioned in Nature Communications. And then, I mean, these are it's just rapidly accumulating. So if you want to, if anybody wants to communicate with me directly, I can try to lead you to the best resources on that. That's probably the fairer way to do it.
Dr. Jones, we're very honored and appreciative that you joined us today. Thanks everybody for coming out, and I hope everyone has a great rest of your Tuesday.
Okay, thank you.
Bye, everybody.